The overall goal of this procedure is to measure the effect of phenotype modulators such as host gene silencing or the impact of small molecules on Mycobacterium tuberculosis intracellular replication. This is accomplished by first dispensing small molecules in 384 well plates or transfecting cells with siRNA and transfectant complex. The next step is to infect cells with green fluorescent protein expressing Mycobacterium tuberculosis or GFP-MTB, add cells to the small molecule containing wells, and incubate the microplate for 5 days. For the RNAi approach, add cells onto RNAi transfectants complex containing wells, incubate the microplate for 3 days, and then infect cells with GFP-MTB expressing Mycobacterium tuberculosis. After staining the cells, the final step is to read the plates using an automated confocal microscope. Ultimately, automated fluorescence microscopy, followed by image-based analysis, is used to measure the changes in GFP-MTB intracellular growth.
The main advantage of this technique over existing methods, like colony-forming unit counting, is that it spares long incubation period and allows more throughput. Demonstrating this procedure will be Christophe Queval, Romain, and Vincent Delorme, 3 postdoctoral fellows from my research team.
The protocols for siRNA library screening and compound library screening utilize 2-week-old GFP-expressing Mycobacterium tuberculosis H37RV cultures. To prepare the GFP-MTB